on either in Boston or on their way to Boston. I'm not going to make it here. Two things before we get started. One is, hey Kim, the CBAC, running it, giving me a hard time. And I want to thank her for all of her contributions. So I think we're all indebted to her for running it. That's one. Okay, so that's one item. The second one is she's moving to your side.
Yeah.
The other is a sadder piece of news, and this is David Rosenbaum, who gave our seminar on October 31st, passed away. And I don't know if you know, but when he was here, he was already very ill with pancreatic cancer. And him and his wife spent the weekend with us. And there are two things that he wanted to do when he came here, because he was trained here at WashU. And the two things he wanted to do, and of course we did everything he wanted to do, was to go to the victory parade of the Cardinals, which happened to be that week, and Ted Drew's for ice cream. So we did both. And David passed away. The funeral is tomorrow. And I talked to Arthur about this, and we're dedicating this seminar to David. So now to the matter at hand, and this is Arthur's presentation. So Dr. Wilde is a friend for many years, and I think everybody here knows of him and his work. Many of you have met him before. Arthur heads the cardiology experimental and clinical activities at the University of Amsterdam, and has really been a pioneer in the application of genetic considerations to the clinical care of patients and connecting genetics to mechanisms of cardiac arrhythmias. Before that, he's done a lot of work on basic cardiac electrophysiology, and we were reminiscing today about Nagoya as one station in his adventures, and then Andre Kleber in Bern was another mentor that he worked with. And now he turned really the University of Amsterdam is to a mecca of what people consider the place that leads the efforts in the genetic basis of the both clinical and research of cardiac arrhythmias and cardiorhythmia mechanisms. So thank you for coming, and it's all yours.
Thank you very much, Yoram. It's a big pleasure to be finally here. We have been discussing this a while, but I'm happy that it now was possible to come this time. And I'm on the way home. I will visit.
Oh, I forgot to say that Arthur is going to get the Distinguished Scientist Award at Heart Rhythm Society meeting in Boston on Friday. Those of you who will be there, it's Friday, 6:30 in the evening or something like that.
It's the same award that you had 2 years ago. Very proud. So the topic I selected after discussing with Yoram is some of our news work on the long QT syndrome, and particularly beyond the causal mutation. And that relates to the fact that, as we all know, within families with long QT syndrome, with every genetic disease, the phenotype is not similar in every individual. So I have a couple of introductory slides, probably not needed, but the field started in 1995 with the identification of the first two genes for the long QT syndrome, KCNH2 and SCN5A, and this is just a list of the inherited primary arrhythmia syndromes. And in 2012, the list is longer, and every time you make this slide, it's almost outdated because there will be a new one that will be added. And certainly after this HRS, I'm pretty sure there will be a couple of new ones to be added here. So the chances that you do find a gene in the syndromes, one of the issues we discussed this afternoon, is This is from 2006, but the data from up till now will also be released at HRS, but are essentially very similar. If you're dealing with a family with long QT syndrome, the chance that you find the genetic basis, and it's actually the chance that you find it with your first hit, is 90% if you use all the clinical information available. But also in isolated cases, it's still 1/3, and this is a lot in genetic terms, because geneticists do genetic testing for diseases where they have not even a 1% chance to find the causal gene. So in an isolated case with a definite long QT syndrome, it's 30% chance that you find the gene. In Brugada syndrome, in families, it's up to 50%, and this is only SCN5A in our case, because we only screen for SCN5A. Isolated cases, it's up to 20%, and a similar picture is for CPVT with small numbers, but also with high numbers that percentage are very similar. So the long QT syndrome is autosomal dominant, rarely autosomal recessive, genetic heterogeneous with these 13 genes involved, and the data on genotyping you've just seen, and there are gene-specific features that I will not discuss today. And the genes, the first two genes were LQT2, encoding IKR, the gene is encoding KCNH2, encoding IKR, and the sodium channel in LQT3. And in 2012, many more ion channels got involved. And my focus of today will mainly be on IKS and on QT syndrome type 1, which is also the main interest in this institute. Now we know that the QTC differs a lot in genotype family members, but this is a compilation of all the different mutations. So in carriers, there is a distribution of QTC values, which is up in a very normal range, up to a very prolonged range. But also in non-carriers, there is a distribution of QTC values, and a few of them have prolonged QTC. But this is, of course, deals with the different mutations, different gender, different ages, and whatever can you think of. It's a reason, though, to do genetic screening, because otherwise it's one of the issues we discussed this afternoon. You will miss all these normal individuals, carriers of the mutant gene with a normal QTC. And one of the reasons why some mutations might be more malignant than others is clear from this paper from the long QT registry, where just took it as an example, where they nicely identified that mutations in conserved amino acids in KCNQ1 channel have a higher risk for cardiac events in type 1 long QT syndrome. Now this is very basic and not needed for this audience, but the pore of the channel is the region that I will focus on in a minute. Ion channels, or the protein consists of different amino acids, and if you line up the amino acids that are in the spore, you end up with this particular slide for many different potassium channels. KCNQ1 is at the bottom here, and there are many different potassium channels. And the idea is here that if you are at the point, let's say position 314, you see there is a G glycine all the way down in all potassium channels. So there is 100% conservation here, which probably means that this amino acid is critically important for channel function. At the other hand, there are sites where it doesn't really matter. For example, here at position 301, there is an A, a T, S in this potassium, an I, an H, a D, and there are many different amino acids at this position. So changing one into the other is probably not going to make a big deal for channel function. Now, there is a score for that, which is referred to as the Shannon entropy score. That is here. I think it is. Sorry, I go back here. The score is the Shannon entropy score, and the score is 1 if all the amino acids are similar through all these channels, and the lower you get, the more diversity there is in amino acids at that particular position. And the different potassium channel mutations, KCNQ1 mutations, that are available in this analysis are given here in this registry. And then you can subdivide in the channel the mutations which are in the lower tertile with this adjusted channel entropy score, so which are not well conserved. And there are those in the upper tertile which are well conserved. And then if you look at this color, and these two will be grouped in the next slide, the different mutations for this study available are here. I just focused on the area around here in the previous slides, but this is the whole channel. We can do the same thing for the whole channel, obviously. The number of patients with information is here. And then you get this curve, and not surprisingly, the more conserved the mutation, so if you are at the upper adjusted channel entropy score, the more they are conserved, the more likely that these patients become symptomatic in their course of life, in the first 40 years of age. And if you are in the lower tertiles, it's less likely that you get symptoms. There are very specific mutations that we are aware of that are malignant. This one is from Peter Schwartz. The majority of patients are from South Africa, where he's looking after a big cohort with the same mutation, position 341. And what he has shown is that having this particular mutation, and it doesn't matter where you are in the world, but leads to a very significant phenotype. A lot of 80% of individuals does get symptomatic. And if you compare this mutation to all the other mutations in KCNQ1, it's almost a double rate of events. The most recent part of this work also comes from the registry, and that shows that mutation in the cytoplasmic loops of the KCNQ1 channel are apparently the most critical ones. They are involved in the transmembrane mutations compared to the N-terminal and the C-terminal end. But if you subdissect the transmembrane area, then the cytoplasmic loop seems to be the critical area. And these are the mutations right here. They are more frequently associated with events than if you are dealing with mutations elsewhere. So the position in the channel in KCNQ1 is important for in risk stratification. Similar data are available for LQT2. I'll just show one slide here, which is older from 2002. But a mutation in the pore is the critical area of KCNH2 for to be more malignant compared to the N-terminal or C-terminal mutations here. And also for SCN5A, these are data that we have just sent in for, just submitted for publication on 403 or 407 patients with 53 different sodium channel mutations. It's a worthwhile effort. And one of the nice things is that, as you would expect, is that, so you know you have two functional defects in aberrant sodium channel. One is the persistent inward current, and the other is the increase in window current. So you can have mutations that the functional defect is simply not known. You can have mutations with one of the two functional defects, or you can have those with double. And as the data, I'm not going to show the data, but with the double, here are the data with the double functional defect, those patients are most severely affected. The single are here, and the unknown is expectedly to be in between because there might be double or single effect. The position in the gene does apparently not matter for the sodium channel. So determinants of the phenotype in general in the long QT syndrome are gene specific. It matters whether you're dealing with KCNQ1, KCNH2, or SCN5A. In general, core mutations are worse than N-terminal or C-terminal end mutations. And in general, as I've shown you the example of the sodium channel mutations, function counts. The more dysfunction you have, the more serious the phenotype will be, which is a logical conclusion, but it has to be demonstrated. Now the question, and that is the topic of the talk, if you are dealing with one identical mutation in KCNQ1. So this is one family with a similar mutation, and as you can see from these four selected individuals, their QTC is very different. This male has a QTc of 436, and this male has a QTc of 509. This is baseline QTc. This male, a nephew of him, has 557, and this female has 467. So what explains the fact that in single family, identical mutation, there is so diversity in phenotype? So why is disease severity so variable in individuals carrying one identical genetic defect? Now, there are a number of reasons, and some of them are easy. Gender and age are known to be important here. The use of drugs is obviously a candidate. If you are using a QT prolonging drug, you will end up with a longer QTc. In females, the interesting thing going on now is the use of anticonceptives, of the hormonal status. It may well be that in females, at one point in the cycle, the QTc is longer or shorter than at another point in the cycle. We don't have accurate information on that, but I wouldn't be surprised. And related to that is the role of anticonceptives. And other genetic factors, and I will focus particularly on this last part. Just to show you some examples that gender counts, females and males in the long QT syndrome type 1 and 2. And age is in the same graph, but as you can see, when you are a male and young age, you are more serious at risk for events than, and at older age, it's females more serious at risk. But as I said, I will focus on other genetic defects. And that is a long list again. First of all, it can be mutations, other mutations in other long QT related genes. It can be more frequent occurring single nucleotide polymorphisms in long QT related genes, or it can be SNPs in other genes. And there are other mechanisms as well. I'll give you some example of each. This is the role of compound mutations. More than one mutation in two different long QT genes. That is what is the definition of compound mutation, and that's a common cause of severe long QT syndrome. So that would be a reason to screen not only one gene, but more than one gene in the case of a severe case. And I don't think it makes sense in a benign case. But in severe cases, we use to screen more than one gene, even if we found one of the given genes. The role of common variants, single nucleotide polymorphisms, is illustrated in the next couple of slides. We have mutations, and mutations are rare, but have a high prevalence. So the chance that you have a phenotype of a mutation is relatively high. You do have SNPs at the other end of the spectrum, which are common, and common means can be 20–30% of individuals could have a SNP, but the penetrance is generally low. But these can influence each other. So there is a chance that the presence of the SNP influences the mutation. And that would lead to the potentially following hypothetical situation. Here's the long QT syndrome patient. Loss of reproduction reserve, or QT interval, is here on the y-axis, and hypothetically, I draw the line here that you become symptomatic if you cross the line of 40% reproduction reserve reduction. So this patient with a mutation is just under that line and will not be symptomatic. If you have a single SNP, you will not be symptomatic. But you guess what the next bar will be. If you have both, you can just cross the line and become symptomatic. And every other possibility is there. Now, there are a couple of candidates already in the literature. NOS1IP is one of them. We still do not know exactly the mechanism, but NEG was done on QT interval in normal population shows that NOS1IP is a hit. So variants in this region are modifier of the QT interval. And Schwartz group was the first to show that it's also a genetic, not unexpectedly, a genetic modifier of the long QT syndrome, and he did so in the same KCNQ1 mutation. In a more diverse population of long QT syndrome patients, a priori showed the very same thing, that polymorphisms in this particular gene modulate the QT interval duration and the risk of arrhythmias in the long QT syndrome. And this was not one mutation, but many more. The effect, though, was very small. this is the 3 SNPs in that area of NOS1AP, and the major allele is here, the minor allele is here, and as you can see, the effect is in the order of a few milliseconds. So a few milliseconds increase in QTC interval lead to a little longer, and only here it's statistically significant, leads to a little longer QTC interval. They were also able to show that one of them had impact on symptoms, so event-free survival was a little higher or a little lower, more events in carriers of the minor allele. So this might impact on the phenotype. But what about the other genetic factors? And then we need to make one step, that you have to go from the gene to the protein. And there are 2 processes involved here. First of all, there is a transcription process, which brings you from the DNA to the messenger RNA. The messenger RNA is taken out of the cell, and translation process brings you from the messenger RNA to the protein. So that is schematically given here. The DNA is up here. The transcription process makes from the DNA the messenger RNA. The messenger RNA is transported out of the cell into the cytoplasm, and in the ribosomes there is the process of translation. And in this process of translation, the microRNAs become involved. So this is the symbol I will use for microRNAs, and these microRNAs may situate be somewhere here on this messenger RNA and impact on how much of the protein is formed. And that may modify whether microRNA binds or unbind there, may modify protein production, and that can go either way. It can be increased protein production, and it can be decreased protein production. Now, the potassium channel is the result of 4 different units. So this is a single gene product, and 4 of these gene products form one functional potassium channel. And I've seen several times these pictures today, so I'm not going to go much more detail. Now, in the normal situation, you have 2 alleles. And the messenger RNA transcripts from the normal KCNQ1 allele and from the normal KCNQ1 allele have their produced protein, so translation process. And in general, all the proteins will be normal. No mutation here, no mutation here, so you only have normal potassium channels. Now I introduce a mutation in this KCNQ1 allele. And that will lead to a not functioning protein, an aberrant protein. They will come together, but it's just a matter of chance how they will come together. So there are essentially many possibilities how they will come together. And if this is an autosomal dominant, if this is functioning as a poison protein, then this channel will not work, and all the other channels will not work, and the only one that will work will be this one. And you will end up with 16 different possibilities, and only 1/16 of them will have normal function. Now, the hypothesis here is that variability in disease severity is caused by an allele-specific translation of the healthy versus the normal KCNQ1 messenger RNA. So what I'm saying here is that if this allele is translated less, then you have less aberrant proteins. If it's the other way, you have less normal proteins. I'll show you that in detail. So let me go back to the. In this case, both are transcribing equally, and you get the situation I just described. Now this one is not functioning too well. Then you will end up with only translation of normal protein. You can also go the other way. You can go the other way. You can only translate the aberrant protein, and you will end up with a phenotype with a longer QTC, with more arrhythmias and more symptoms. The point now is that in the area where these microRNAs bind, the 3' UTR area, the messenger RNA, this is enlarged here. This is also a coding sequence. So, and in the coding sequence, there are potentially SNPs, single nucleotide polymorphisms. And in KCNQ1, there appear to be 3 polymorphisms that are relevant here. And I'll show you in a minute why they are relevant. Now, the messenger RNAs bind to this area. And this area, as I just said, have a coding sequence, and there are SNPs, and they might modify the way these messenger RNA bind, and the strength they bind, or maybe even unbind completely. And if that modifies the production of this protein, and you might even end up with proteins that are completely destroyed. So there is a coding sequence here on the 3' UTR area, and the messenger, the microRNA, is able to bind to that. And the same holds for the other thing, for the other 3' UTR area, and the microRNA binds to it. Now I introduce a mutation here. This A is changed into C, and that might impact on the way that the microRNA can bind here. So it can unbind, or you can create a binding site. It can go both ways. So let's focus on this particular variant in the 3' UTR area. It's an A. The major allele is A. It's similar in the normal allele and in the abnormal allele, which means that you get equivalent messenger RNA processing from the normal and the mutant transcripts, right? So this is the situation here. Now I move into the situation that you have a variant here, the G, and the variant enables the microRNA to bind, which suppresses translation of this production of this protein, and you end up with only normal channels. And at the other side of the spectrum is the variant is on a normal allele. The microRNA binds here. You end up with suppression of production of the normal protein, and you end up with more aberrant transcripts. So at this side of the spectrum, you have less QT prolongation. At this side of the spectrum, you will have more QT prolongation. So that has to be tested, and that was tested in 168 patients with long QT syndrome type 1. They were equally divided between our center and the Mayo Clinics. So this was a collaboration with Mike Eckerman. And the regular clinical characteristics of the patients are here. Probands, family history for sudden death, syncopal events, and documented those out. It was all recorded and was equally in the two groups. Now what you have to do now is, and that is critical in understanding this, you need to know where on the 3' UTR area these variants are. So you need to know the allelic location of the SNPs. Are they on the same allele as the mutation, or are they on the other allele? And this is an example in the pedigree. The father here is affected. So this is the mutant allele, and the 3' UTR genotype is here, GAA, and the normal allele of the father is here. Now his affected son has on the normal allele the G and the G. They can't come from the father, because that was genotyped differently, so they should come from the mother. And the same here, these minor alleles are coming from the normal allele. So in this case, the aberrant function is coming from the normal, is associated with the normal allele. And this is the data for one of these SNPs. It's on this particular position. And as you can see, if that SNP, the major and the minor allele, is similar on the mutants and on the normal, we have in our population in Amsterdam, we have a QTC which is around the high to normal value. If the minor allele is associated with the normal allele as far as the mutation is concerned, we get less translation of the normal protein, more translation of the aberrant protein, and you do expect a longer QTC, and that's right what we see. if the situation was opposite, we could not study in Amsterdam because we simply had no patients with the combination of the mutant allele and the minor 3' UTR allele. But in the Mayo Clinic group, that information was abundantly available. There were 31 patients with the minor allele on the same allele as the mutation, and you saw less transcription of the mutant allele and normalization of the QT.
Was there something systematically different in your methodologies that gave you differences between the two clinics?
No, this is just
Controls?
this. You mean the difference here?
No, between Amsterdam and Minnesota.
But
It's like the QTC and the controls is, I guess not.
No,
It's 460 in Mayo and
scale is- the
440
scale-
for
The
you.
scale's different.
the scales
No,
are
but it's
different.
440 for you and 460 in Mayo.
So it's
No.
for 55 here and for 40 here.
Okay.
Yeah, but we are not talking about the same mutations here. They are different.
Oh, okay.
But the point here is that when the minor allele is associated with the normal variant, you have a short QT in both groups. When it's the opposite, we have no data there, but it's associated with the normal.
There's multiple different mutants that you're comparing.
Yeah. And different between the two centers.
Okay.
Now, so this is just one SNP. We have data on 3 SNPs in the 3' UTR area, but the results are essentially different. So when the minor allele is with the normal variant, you have the longer QTC. This is relevant here, this is relevant here. And when it's at the opposite side, when it's a mutant allele, you end up with the more normal phenotype. QTC is reduced. Now that is obviously something you can show on the whole level of different mutations, but it should also be true in the family that I was referring to. And here is the individual with the pretty normal QTC, and he has the equal distribution of the translation process. But here's the important one. Here's the one with the very long QTC, and he has a reduced translation of the normal protein because the minor allele is associated not with the mutation. And the same is relevant here. So in individual families that are big enough to show the effect, you can show very similar behavior. What about symptoms? We have to group the 2 populations to show the effect for symptoms, but it coincides with QTC as you would expect. So when the minor allele is in front of is associated with the non-mutant channel, with the non-mutant gene, then you have the most symptoms. You have the longest QTC, you have the most symptoms. And if you are at this side of the spectrum, if it's in front of the normal mutant allele, you have less translation of mutant protein, you end up with relatively benign phenotype. We did some basic work behind this clinical associated work to show that there is indeed translational repression by the minor allele in 2 different cell systems. And I'm not sure you can see it, but the different, so there are these luciferase experiments in different cell types. On every allele, the G is engineered here, the G is engineered here, the A is engineered here, and always there is less, about 25% less production of the luciferase in different cell experiments. And on the messenger RNA level, we could also demonstrate this is a tissue, human tissue, that if there are patients with a G allele in front of the normal, then you do get a little less. It's not much effect, but it's a significant effect of messenger RNA level. And this is all just published in the European Heart Journal. And that then brings me to the conclusion that SNPs in the 3' UTR area of KCNQ1 repress translation, and heterozygous carriers made us have allelic imbalance. And allelic imbalance becomes relevant when one allele contains a mutation. It doesn't make a difference when both alleles are the same. when one allele has a mutation, it may become clinical relevant. Maybe not only on QT, but in every other disease entity. And in this disease entity, the allele-specific repression has an effect on QTc and symptoms in case you have Q1 mutation carriers. This, I think, is a very nice story to tell, but the work has actually been done by one of the Ph.D. students that will defend his thesis. In a month from now, Anke Thijs has been instrumental, and Ika Pinto in our institute, and the collaboration with Mark Eckerman is long-standing and appreciated in this sense because these data were needed to get this to the complete story. So thank you very much for your attention.
Thank you, Arthur. Questions?
So that's really nice allelic analysis and the non-coding regions, and it suggests that actually all the variants, or a lot of it, could be within the gene itself. Are there any polymorphisms in the same genes that could be, and have you done the analysis to show whether common variants are affecting the disease mutant effects?
There are common variants in these genes, but they do not, as far as I'm aware, modify the phenotype. No, not in great detail. So the NOS1AP data that I showed you is variants in other genes. They do have a modest effect, but it's in the order of a few milliseconds.
But mutations within, or polymorphisms within the gene itself, if they were just looked at, let's say, by recombinant analysis, people might not actually see anything because there might be no change in properties, but if they had an effect on protein expression.
But you know, this area is important for protein expression. The role of microRNAs relates to the 3' UTR area, and that is where the protein expression is actually determined.
Coding variants can also have a big effect on expression because they can have effects on trafficking and
but we haven't looked at it.
So for those mutations that are residing in the 3' UTR area, are they predicted to be any of the targets of any of the microRNAs that are known?
Yeah.
And
So it's 378, microRNA 378 is the microRNA that we think is related to this.
and if you do a in vitro assay to overexpress these microRNAs, do you see the same effect?
So we haven't done that yet. We are actually, this is one of our targets for the stem cell studies, because that's where you easily can do this type of work. And you can recapitulate or recover the phenotype, or rescue the phenotype. It's also a potential target for treatment, because if you know your patient has this combination, and you can modify the translation process by adding microRNA, then you could modify the phenotype.
Very interesting.
For the case when both alleles are the major alleles, are they really translated equally, or is there any role for genomic imprinting in these genes?
We don't think so. I don't have the arguments now.
Is that based on the functional distribution of the dominant negative mutations? Or?
Yeah, I don't have the argument.
But there could be an effect that's not genetic, but that the stability of the protein would be altered, right? And you get a different level just because of that.
Yeah, but you can show in the luciferase experiment that doesn't relate to the KCNQ1 protein, but you see a similar effect. You impact on the translation process, and you simply get less protein.
Can I ask a question about the polymorphisms? Did I understand your answer to Colin's question that there aren't polymorphisms in the sequence that impact?
We haven't tested in
Okay.
KCNQ1, but if they would impact on channel function, they would have popped up as non-QT mutation, I would say.
not if they're very low defect. Very low. But they would have been
No,
detected.
but then they would have been detected with the GWAS for the QTC.
Yes, they would be identified as variants, but
Yeah.
how
I don't
tightly
think-
they'd be associated, I don't know.
That's one of the issues we struggled with, because
Mm.
the prevalence of these SNPs in the 3' UTR area, you can make the same argument. Why don't they show up as a modifier of the QTC interval in the normal population?
Yeah.
Because they will reduce KCNQ1 protein.
Yeah.
But apparently the abundancy of all these potassium channels is such that you can do with 10% less KCN protein. In the
But there
normal
must be
situation.
some effect.
In
There
the
must be
normal
some
si-
effect,
yeah,
but it may not hit the threshold
yeah,
in the
we didn't-
GWAS.
We don't find it in the, yeah, but you also don't find it in the, if you measure QTC. The effect is apparently then too subtle, but it becomes relevant
Yeah, yeah.
when there is a mutation in
Absolutely.
one of the alleles. We tried the same thing in KCNH2, but there are no variants in the 3' UTR area, so I don't think this is a relevant mechanism for KCNH2. It's relevant in SCN5A. We have beautiful data on SCN5A. Hypertrophic cardiomyopathy. It's,
I mean, I actually wouldn't take you anything to go back now and look at the polymorphism status of those individuals, right, and do the same analysis effect.
yeah, we will do.
Okay, any other? And thank you again, Arthur.